Okay. Well, good afternoon, everybody. Welcome to this ERKNet ERA seminar on podocytopathies. My name is Tom Nijenhuis. I'm an internist nephrologist from the Radboud University Medical Center in Nijmegen. And it is my pleasure to introduce our speaker of today, which is Professor Tobias Huber, who is the chair of the Center of Internal Medicine and director of the Third Department of Medicine at the University Medical Center Hamburg-Eppendorf. Together with his team, he is internationally recognized for discovering signaling pathways relevant for kidney development, filtration, maintenance, and disease. And he has published over 280 articles, I think, already. Is one of the internationally most cited podocyte researchers and received numerous national and international recognitions, including the Donald Selden Young Investigator Award of the American Society of Nephrology. And he is an elected member of the American Society of Clinical Investigation, the Association of American Physicians, and the National Academy of Sciences. And a major focus of his career has been to mentor, educate, and facilitate careers of young talents and clinician scientists. And recently he was actually elected as the inaugural president of the newly formed International Society for Glomerular Disease. And before I give Professor Huber the floor, first I would ask everybody to ask their questions in the questions box, so that after the presentation we can have a nice discussion on all the facts that Professor Huber has given us in his presentation. Professor Huber, the floor is yours.
Tom, thank you so much for the introduction, and thank you very much to the ERKNet and also the ERA team for having invited me, and thank you all for listening and participating. It's a real pleasure to be part of this educational webinar series, and I think we all across particularly Europe benefit from the ERKNet and from the interactions that we gain through ERKNet, including the patient representatives that are participating in ERKNet. I've been asked to talk about the physiology of podocytopathy. I will give some general introductions and general considerations of podocytopathies, and then I will kind of exemplify ongoing dynamics in the field with latest research from our teams in Hamburg. Most of the work has been published within the last year. Now, let me start with a few general considerations. I will start with 7 general considerations which should be considered important in understanding podocyte diseases. First of all, evolutionary, the glomerular development has been a key prerequisite for the development from maritime life to terrestrial life, and actually as was the capability and ability of excess filtration that allowed life to adjust to different environments. It might not be exaggerated to say that glomerular and filtration development has been a key step in the development of human being. This was number 1. Number 2, the glomerulus itself is being composed of different cell types. It's a highly intricate and complex architecture structures being composed of not just, of course, podocytes, mainly the capillaries being covered by podocytes and the mesenterium, as well as the periapital epithelial cells. And throughout the last decades, we learned about the importance of these cell types and how they act together. Third, which is really important, while research, particularly 20, 30 years ago, mainly focused on the individual function of these different cellular compartments. We learned over the last 15 years that all of these compartments form almost like a functional syncytium, are closely interacting, are mediating signals to each other, and there is no sole disease of a podocyte or mesenterial cell or PEC cell or endothelial cell or even a tubal cell. All and everything is all the time connected, and injury of one of these compartments is always affecting the other compartments. Fourth, from the biology, the filtration barrier is one of the most complex structures in the human body. It's composed of the endothelium with the largest fenestra of a human body, And this endothelium is covered by the thickest glycocalyx in the human body, which is followed by the thickest basement membrane of the human body, which is followed by the most specialized epithelial cell in the human body, the podocyte. And the podocyte foot processes are being bridged, as seen here, by the most complex and longest cell-cell contact of the human body. So evolution really truly invested its best and most sophisticated parts to ensure filtration. And the filtration is being based on the fact that these capillaries are pulsating with a pressure, with an unbelievable pressure of 50 millimeter mercury. So there's no other capillaries in the human body being exposed to such a high pressure. Interestingly, in all the Chimaela diseases, and this is, the ultimate goal of cellular research in the Chimaelas, is understanding Chimaela diseases. And the reaction patterns throughout these cellular compartments, it's quite uniform, regardless of underlying injury signals. For example, for the podocyte, the key reaction patterns are foot process effacement, simplification of the cell body, eventual hypertrophy of a cell body, and ultimately loss of adhesion and depletion of podocytes. For parietal epithelial cells, it can be proliferation of these cells, like in crescentic glomerulonephritis. For mesangial cells, it can be mainly excess matrix production and deposition and proliferation. And for the endothelial cells, it can be proliferation or swelling. And all these patterns appear at a different scale in different Chimaela diseases. Another thing which is important with all Chimaela diseases is the site of injury. So whenever we see large-scale proteinuria, nephrotic range proteinuria, this always means that the outer part of the filtration barrier needs to be damaged. So there is no nephrotic syndrome without podocyte injury. Whenever we see, on the other hand, a nephritic syndrome with erythrocyte casts, for example, then always all layers of the capillary and effiltration barrier need to be injured, often, for example, by deposition of immunoglobulins underneath the endothelial compartment. Moreover, there is usually a sequence of Chimaela injury to CKD. So don't forget, when a Chimaelus is getting sclerosed, this also affects the peritubular perfusion, blood perfusion of the nephron, which is highly oxygen dependent, usually leading then to a loss of nephron. This means injury to the Chimaelus, subsequent total scarring of the Chimaelus leads to increased fibrosis, loss of nephrons, and CKD. Now, let's come to the next steps and come to the main function of the Chimaela filter, which is obviously filtration. Whenever filtration, and a key function of filtration is really to have a high hydraulic conductivity while maintaining large proteins, of course, cellular components within the circulation. Therefore, it's important to understand what's happening in proteinuria, which is potentially a life-threatening condition. Now, earlier studies, when the slit diaphragm was discovered by early EM work by Marian Farquhar and George Pallada in the 50s, they described it as slit diaphragm because they observed a thin electron-dense membrane, and until today, the textbook keep mentioning it as a slit diaphragm, a thin membrane. This doesn't make any sense. If all this filtrate of 140 to 180 liters are passing the structures, by no means this can be just a thin membrane. The truth is, and this is something that we found by the first cryo-electron tomography of native unfixed kidney tissue, is that the slit diaphragm is just and fixation artifact. So whatever you see is the diaphragm in electron microscopy is due to fixation, and then basically gluing the layers of the slit configuration together. So what it really looks like is shown here. It's a multi-layered structure, and these structures are highly flexible, which we found by modulation and animation of these molecules in the slit, like nephrine. And we also found that they are non-restrictive to albumin. So albumin can actually pass through the slit diaphragm. Now, this in turn means that the basement membrane needs to be the major structure this holding the passage of albumin. Now, one of the current theories, which is coming out of Thomas Bensing's lab in Cologne, which might really best explain how the GBM is actually retaining and being the filter while the podocyte is important for GBM function, is that the pressure of the foot processes on the GBM keeps the GBM matrix in a composition that makes it restrictive for albumin, while, as our studies were showing, the diaphragm is non-restrictive to albumin. However, podocyte injury, foot process effacement leads to less pressure on the GBM. The GBM is losing its texture, and by that, it's getting permeable to albumin, and this in turn causes proteinuria. Now, if the slit diaphragm, as we found, is non-restrictive to albumin, then of course the question is, what are the functions of the slit diaphragm? And this is something which we suggested many, many years ago, that the slit diaphragm complex is bound to a highly active signaling machinery on its inside, regulating the ectocytoskeleton, cell survival, transcriptional programs, cell polarity, endocytosis, also calcium signaling. So it's actually a signaling platform to maintain the glomerular filter. The other thing that we proposed also 10 years ago already is that the podocyte slit diaphragm is a unique cell-cell contact. It cannot be described as a tight junction, adherent junction, or gap junction, or a neuronal synapse, but it contains components of all these cell-cell contacts of neuronal synapses, tight junctions, adherent junctions, and gap junctions. So it clusters all of these components while it forms really a cell-cell contact of its own. Now, as promised, I would like to show where this field is moving with current work. So it's very important to understand the composition of the slit diaphragm. Many discoveries have been made on the basis of genetic studies, monogenetic diseases causing proteinuria and coding for components of the slit diaphragm. But we didn't have a comprehensive overview of all components. And this is recent work led by Florian Kramer from our team and Bernd Fackler from Freiburg, where they for the first time applied multi-epitope mapping using multiple antibodies against slit diaphragm components as control, using the knockouts for these components. By that, being able with different buffers and solubilizing the plasma membrane to really, for the first time, purify the native slit diaphragm complex, which was amazingly interesting to understand how this slit diaphragm complex is being composed. And to our surprise, it's really being composed of structural components, but also of lots of ion channels, single transmembrane signaling molecules, non-receptor tyrosine kinases on its surface on the intracellular side. And all these complex machineries are building basically a functional complex, which is sending signals also in response to filtration. Now, to prove these components and their functionality, we used the Drosophila nephrocyte system, and we did knockouts in these systems and could show that these components are required for the maintenance of a filtration mechanism, at least in the highly conserved Drosophila nephrocyte. Now, moving from this recent work to the next aspect, and this is mechanosensation in the cumulus, which is becoming so important because we learn the new medicine and medications that we now use for all cumular diseases, like SGLT2 inhibition and since 20 years, RAS blockade. They all work probably through lowering the intracumular pressure. So the intracumular pressure is being built up by the blood through the afferent and the efferent arteriola. The wider the efferent arteriola, the closer the afferent, the lower is the intracumular pressure. And oppositely, if the afferent arteriola is wide, the efferent is rather narrow, then we have an increased pressure. This increased mechanical pressure in this high pressure capillary system is a main driving force for CKD, for cumular disease progression. That's why medications lowering the intercumular pressure are becoming the main medications to slow down CKD and cumular disease progression. Now, this is basically entertained for SGLT2 inhibition, and you know all the tubular cumular feedback, but you inhibit salt and sugar reabsorption of proximal tubule. More salt is appearing at the distal tubule. This is sending signals from the juxtacumular apparatus and macula densa, and then it's basically affecting the efferent arteriola, and by that it's directly affecting, it's widening the efferent arteriola, by thereby lowering the intercumular pressure, and by that basically relieving the cumulus from pressure. And this is certainly one way how SGLT2 inhibitors work. Now for us, and now we come to another 23 recent publication from our teams in Hamburg, but we really ask us now the question, what is the intracellular sensing of this mechanical pressure? So why does relieving the mechanical pressure is being the main concept of slowing cumular disease progression? So we used basically a model of early diabetic nephropathy, where we don't see any structural or cellular changes, but where we have a hyperfiltration. So increased mechanical pressure, and then we did bulk RNA sequencing and single RNA sequencing throughout the cumular cells and compartments. And we found a series of transcription factors significantly being upregulated in hyperfiltrating early diabetic animals. And mainly upregulated were factors like MRTFA, B, but also YAP, TAS pathway, all mechanosensitive pathways. So basically the hyperfiltration leads to a strong upregulation of mechanosensitive pathways. Now to see whether this is anyhow connected, because this came from an early diabetic model to diabetes, we also did an artificial ex vivo system where we removed the kidneys, perfused them with high pressure, and again analyzed the glomeruli and could see the exactly same pattern of transcriptional activation. Now, this is basically now the novel model, which might explain the beneficial effect of all the things we are doing to slow down CKD progression over the last 25 years. 20 years RAS blockade, last 5 years SGLT2 inhibition. And basically what it does, the messenger cells, but also other cells in the cumulus are sensing mechanical pressure. They translate this mechanical pressure into activation of MRTF transcription factors. And this in turn directly leads to an increased extracellular matrix production and is a strong pro-fibrotic stimulus, at the end leading to the cellular changes that we observe, for example, in diabetic nephropathy, showing the molecular basis behind lowering intracumular pressure. Now, let's move on to other key aspects of podocytopathies and cumular disease. Of course, you all are aware that genetic diseases are important. Work that nicely summarized this by Jeffrey Cobb, Paola Romagnani, and many other authors in the field, showing how important monogenetic diseases are, particularly in early adulthood. The older our patients get, the fewer monogenetic diseases we see, but genetic modifiers maintain very important for susceptibility, and we are still learning a lot about genetic modifiers. I would like to give you a recent example of a new gene that we discovered. So we talked a lot about the GBM being very important for proteinuria. So we got very interesting whether we can, for the first time, identify GBM modifiers. So most genes, over 50, that are linked to monogenetic diseases of proteinuric diseases are being directly linked to a podocyte through all the compartments like diaphragm, adhesion, cytoskeleton, mitochondrial, metabolic mechanisms. And so we were looking now whether there are eventually genes can be found that also modify the GBM. And by screening through a large panel of families, by genes that we looked at that could be potential candidates because they are highly expressed in the podocyte and they are known to modify the GBM, we identified a family with the gene P3H2, which is an collagen hydroxylase, and we did the knockout model. And when we did the knockout, you can appreciate the collagen 4 staining, that the GBM is irregularly structured in contrast to the controls. And we see substantial change in the collagen network. And to break a long story short, because it's been published last year, and again, many people from our teams in Hamburg have been involved. The lead was Florian Krahammer. We basically come up with a novel concept. It's not just podocyte structural genes being mutated, leading to podocytopathies, but it can also be enzymes that help processing collagen variants. And this is a hydroxylase, which is important for the correct folding of collagens before they are being secreted and being deposited in the GBM and basically assembling the GBM. Now, lack of this gene or also acquired impaired function of these genes, like in diabetes and other diseases, might impact the correct assembly, leading to proteinuria. However, it's not only just the new genes that we need to care about, it's also long-time existing gene programs that need to be revisited with new techniques when we can better understand the mechanisms. As a proof of concept, I would like to give another example, which just actually appeared online yesterday from our team. And this deals with Fabry disease. You've all heard about Fabry disease, a lysosomal storage disease. It has been known for decades. It's actually the first disease where an enzyme replacement therapy was established. So you give IV the lysosomal enzyme, it somehow enters the cells and restores in part the lysosomal function. And one cell that is mainly affected, while all tissues can be affected, is the podocyte in the glomerulus, forming these zebra bodies, leading to the swelling of these podocytes and also a glomerulopathy. So something that was really, and this is the actual paper, many people have been involved, were led also by Victor Puelles, Christoph Schell from Freiburg, and Fabian Braun. Now, basically, what we saw is when we looked at, and many collaborators across the world, and I apologize for not all of them being mentioned here. Now, by looking on human samples that have been treated with enzyme replacement therapy, we observed that while the depositions of GB3 as an end product of alpha-galactosidase are being diminished, the podocyte architecture is not improving so much. So this basically let us speculate. It's not just about the end product GB3, but that there might be an aberrant signaling in this lysosomal storage, which we need to discover. So this was our hypothesis. So loss of alpha-galactosidase. Yes, it's known it leads to lysosomal dysfunction. Yes, it leads to GB3 deposition, but we speculated that there might be other distinct pathways leading to an endosomal disruption and cellular injury. And by that, it's very important to revisit the physiology of the lysosome, all these transporters, all the molecules being processed. And we in fact started doing a lysosomal proteome analysis, looking for lysosomal proteins by silicate labeling these proteins in a Fabry knockout model versus control models. We could also look for the half-life of proteins, potential accumulation, and we tried building up protein networks. And it's interesting, there was kind of a distinct protein network that is significantly impacted in Fabry's disease. And among these 8 top regulated proteins, one catched our eye particularly, which was synuclein alpha, which seemed to be in a keynote for down and up regulated proteins. So very interesting protein. So we got very interested in this gene, and we looked, of course, also confirmed it on protein level, not just proteomics level, but also antibody-based level. And then we went back to human patients and we looked for alpha-synuclein, and we saw that in untreated Fabry patients, we see a dramatic accumulation of alpha-synuclein, particularly in the podocyte compartment, which wasn't improved so much after enzyme replacement therapy. So fitting our theory that the podocyte doesn't improve so much, and in fact, this therapy is just slowing but not reversing or holding the disease. So alpha-synuclein became an interesting candidate, and of course it's also known from as a molecule that causes in the neuronal system Parkinson when being deposited. Now we did a functional experiment by knocking down alpha-synuclein very successfully, and then we looked again on the lysosomal phenotype in Fabry cells. And it's interesting, then you see controls, you see Fabry cells, and you see when we take out alpha-synuclein alpha, we can restore the lysosomal dysfunction or enlargement. We can partially restore the lysosomal pH, and we can partially decrease again the lysosomal ROS production, which happens in Fabry cells. So very interesting that alpha-synuclein seems to be a product of Fabry's disease, but also a driver then of further cellular injury. Then we used, together with the Broad Institute in Boston, so-called connectivity mapping that identifies transcriptional programs that are being up and down regulated, and then in certain cells. And we used our Fabry cells, and we looked whether there are, to explain this better. So what the Broad Institute did, it used all medications on the market and looked what transcriptional profiles they do. Then you can manipulate the cells, like in doing Fabry's disease. And then you look how this is altering the transcriptional level. And then you know how all different drugs are impacting it. And then you are looking for things that do exactly the opposite. Basically, Fabry's disease is causing upregulation of a transcription profile, while you found one medication or drug that does the opposite, and then there's the hope that they both being given together might ameliorate the phenotype. Now, by looking at this, we found a very strong signal for beta receptor agonists. So if we applied beta receptor agonists to the cells, we could completely diminish the alpha-synuclein accumulation. Dramatically interesting finding. Now, there was a very recent, or not very recent, but recent paper coming out on Parkinson where they screened in Parkinson. Remember, it's a disease where alpha-synuclein is deposited. And they found, again, that beta-adrenoceptor agonists are decreasing alpha-synuclein. So this was a nice confirmation. And now to summarize the story, so basically, it's not just about identifying new genes for podocytopathies, but also coming back to better understand already existing and known genes with the new techniques like single cell techniques, connectivity mapping, complex AI-based computational analysis. And by that, we found that the mechanism of alpha-gal loss is an accumulation of alpha-synuclein. We found how this is being regulated, and we found potential new ideas for additional treatments on top of ERT. If you are interested in the theme of Fabry's disease, and ERKNet is a network of rare diseases, the Worldwide International Meeting on Fabry's disease next year, and it's all just every 2 years, but next year it will be in Hamburg. We will be hosting the meeting. Remember the date, and you're all being invited to join this meeting. Now let's come back to a few more examples of disease and come back to what we call primary podocytopathy. So what we name primary podocytopathies are diseases, not systemic diseases that are affecting many cells and many tissues and many organs, but that particularly affect the podocyte. The three diseases are minimal change, global foot process effacement, FSGS, very similar to minimal change, global foot process effacement, eventually same underlying cause, but also different causes, but here we are losing podocytes, and membranous nephropathy by forming antibodies against podocyte antigens and antibody antigen deposition underneath the podocyte, leading to disease. So what is ongoing new work? Again, another example which just came out from our teams in Hamburg. This work was being led by Nicola Thomas, together with our nephropathologist Thorsten Wiech, and they looked carefully throughout our cohorts on complement activation. It has been known for decades that in membranous nephropathy, complement can be activated, but it wasn't clear which components. So Thorsten Wiech developed an assay where he can screen in patient samples for the activity of different complement pathway, the classical, the alternative, and the lectin-based. And he found that the classical pathway is being activated in all the membranous nephropathy patients, while we also see an activation of the alternative and the lectin-based pathway. And while there is a dominance of IgG4, which is actually not complement activating, we see all other subclasses of IgG, which in turn are responsible for the complement activation. Now, to turn this very recent paper into a short story, using our animal models and combining it with models that were complement deficient, and we could show in Hamburg that basically complement is a part of the injury. If we take out complement components, proteinuria is being decreased, but not completely ameliorated. So the conclusion is that yes, there is a complement component, which talks in favor of the ongoing trials using complement inhibition. And on the other hand, there's also a complement independent pathway, something that needs to be surely further explored. And I know that there are many clinicians under us in this webinar today, and this is how things develop. The treatment for membranous nephropathy will include more and more targeting, of course, of the B cells producing the antibodies at different levels, including anti-CD38, and it will include in the inhibition of different complement pathways. And at the end, there might be even a combined multi-dimensional therapy regarding to the different stages of membranous nephropathy. Let's move on to the last podocyte disease, which I want to entertain today, minimal change disease. We know it's an immune-mediated disease, and there has been a very interesting paper coming out actually last year by Astrid Wein's group in Boston. And Astrid Wein's team discovered that in a third of the patients, antibodies to nephrin in minimal change disease can be discovered, which is very interesting and which puts the minimal change disease basically on the menu of potentially antibody-mediated podocyte diseases. While we see in membranous deposition, we don't see a deposition in minimal change because these antibodies are directly binding to the functional component of the slit diaphragm, leading to changes of the slit diaphragm, leading to an increased permeability of albumin and proteinuria, and being a potentially quickly reversible disease. It's not quite easy to detect for antibodies. The ELISAs are using lots of amplifiers and are not working on a regular basis. So for that, in Hamburg, we came up with our team with a new strategy of IP, basically using nephrin to IP the serum of patients to find whether there are anti-nephrin antibodies. And in fact, now for the first time, we have a very robust assay to detect circulating nephrin antibodies, which is very interesting. And again, we also see it in almost up to 40% of minimal change diseases. We don't see it in IgA, we don't see it in membranous, just one case. We just see it in one case of FSGS, and we know there's a transition from minimal change to FSGS, but we really see it particularly also in relapsing FSGS. So this is very interesting, and this opens new avenues to better understand the pathophysiology, but also use it as a novel tool to monitor disease. And here now, as we did it for membranous nephropathy in Hamburg, we would like to ask you all watching us today in the network, we have, please feel free to contact us, because we have the national Hamburg GN registry, all ethics are in place. Send us the serum of patients. We detect for you nephrin antibodies in minimal change disease. We can send you result, but of course, based on our ethics, you're not allowed to use it for treatment decisions, while it might be interesting for you to know if you're considering somebody frequently relapsing minimal change patient with rituximab. It is an interesting knowledge, while you're not officially allowed to use it for clinical decisions. But you also help up to build our registry so that we can learn quickly in the follow-up how these patients with observed nephrin antibodies react to therapies, how the outcome is, and so on and so forth. So please send us your samples and feel free to contact us. So I'm coming to the summary and outlook. I hope I could entertain you the complexity of the Chimelia filter. Most amazing structure in the human body, built up by highly specialized structures, and the podocyte forming the outer part. It's being diseased in many diseases, genetic and acquired diseases, which we called podocytopathies. There is a very interesting and exciting renal discovery path. For us, everything is being based on registry that we use. The techniques today allow a higher molecular, spatial, temporal, functional resolution, really leading to novel insights on glomerular diseases and podocyte diseases. It's very important that we together collect samples to discover these diseases. We need the Hamburg GN registry. And it's exciting times also in terms of involvement of pharmaceutical industry. We have over 60 ongoing trials on glomerular diseases, which have some difficulties in recruiting patients, and we'll come to this in a minute. It's important that we recruit patients to these trials, that we help exploring and help bringing new medications on the market. There are different consortia. I would like to mention the German BMBF Stop FSGS, which is closely connected to ERKNet actually, and involves Greifswald, Erlangen, Aachen, Köln, and Hamburg is the leading site at the moment. You can contact us for cases and research questions. Then you are aware that we have a CRC and SFB on immune-mediated glomerular and rare diseases. And in Hamburg, I would like to thank all the people's work that have been showing, and we clustered now all renal experts in Hamburg in what we call Hamburg Center for Kidney Health. These are 150 people doing kidney research, and they are now being clustered in one new building that we are building, so that all people will work on one big building and laboratory, joining their forces together, and everybody's happy to contact us to collaborate or to join. And then, and I mentioned this before, glomerular diseases are becoming increasingly complex. It's becoming a subspecialty of nephrology, and we need to react for this. We need educational programs. We need clinical trial centers for glomerular diseases. We need fellowships, installed fellowships, and we need worldwide networks. And to achieve this, we very recently, 2 weeks ago, just launched the new society, the International Society of Glomerular Disease, which is actually closely connected to ERKNet, and ERKNet has a sitting chair in our steering committee. And by that, we really, with our pillar committees, we elaborate at the moment how we do the accreditation for glomerular disease centers across the world. We set up educational programs. We professionally support centers of excellence for glomerular disease. We map out clinical trials across the world, and we do a matching of patients to trials. And of course, we also foster and promote translational research. And by that, with this society, we really push forward from the understanding of glomerular disease, transforming it to a novel glomerular medicine and subdiscipline, and by that, hopefully ending up on glomerular health. So I'm happy to serve as the inaugural president of this society, and we will host the first meeting of this society together with the International Podocyte meeting in 2025 in Hamburg. Please all come and join. And this is how quickly increasing our members are. It's more of a global north, but we are reaching out to the global south. It's across 6 continents and many, many countries that are participating and helping us to elaborate. Now, today is your day. Today is your moment, our moment, and I would, whoever's interested, it's cost-free. You can be still part of the inaugural members. Just pull out your cell phones, scan the barcode, and you can sign up as member. Do it right now. Hopefully we get 100 new members in the next 5 minutes, and we together can hopefully move communal medicine forward. With that, I'm coming to an end. I would like to thank Tom again for introducing ERKNet and Franz Schäfer for this wonderful network, which helps us all. ERA, of course, also, which is a joint partner for this meeting today, and IPNA. And I'm looking forward to your questions. Thank you so much.
Thank you very much, Tobias, for this great overview and also introducing some new concepts. I'm looking at the question box over here, and there are some questions, but I would like to motivate everybody to put in new questions. One question is from Jens Koenig, who asks you, would you be able to detect anti-nephrin antibodies in biopsy specimens from a transplant kidney as well, even if the biopsy was performed more than 12 months ago? And do you think it makes sense to look for anti-nephrin antibodies in the patient's serum about 2 years after explantation of the loss of the transplant kidney in a child who has been transplanted for infantile nephrotic syndrome of the Finnish type? So a very specific question.
So very specific, great questions. Coming to your first question, so there is an increased pattern of nephrin signal which can be done on histology sections. Astrid Weins is doing this regularly. We have to say, to us, this is not so convincing. It's not a black and white fluorescent staining, but it might be based on the sample preparation. And in Boston, they use really fresh, cold frozen tissue, and we use fixed tissue. So maybe it has to do something for that. We are 100% reliable now on our new method of detecting the circulation. So and if you send us samples, for now, we just rely on serum samples. This is number one. Number two, would it make sense to somebody who lost eventually the organ after transplant 12 months ago? It makes surely sense if you are considering a retransplantation. So we do this for patients with recurrent FSGS, and we detected we have now one case where we detect nephrin antibodies. Of course, this is a patient that you not just treat with then plasmapheresis, but in addition rituximab in the hope to decline the level of circulating nephrin antibodies. So yes, this makes sense. And then the third question, is there an increased appearance of antibodies in genetic mutations? There has been very few cases long ago with mutations of Finmaya of the Finnish type, where antibodies have been detected even before now the paper of Astrid Weins, certainly not in Finmaya, but in Finmaya where there's some misfolded nephrin eventually giving rise to some more increased antigenicity in antibody forming. But these are very rare cases, and we really don't have enough insights now. If you have a cohort of these patients, feel free to send. We are happy to measure them for you to see whether this is a more common phenomenon. I hope this answers all your questions.
Yeah, we can't know because Ori has to put a comment in, but I'm sure because you answered it very detailed. I was wondering, you showed us the importance of the mechanosensation and the relationship to the GBM, but also the relationship to treatment with ACE inhibitors and the newer SGLT2 inhibitors. And I'm always wondering that, well, these SGLT2 inhibitors, but also these ACE inhibitors, they work with any type of glomerular disease. While there are, of course, different injury patterns, different light microscopic patterns, Is it really one size fits all, or should we look for other types of therapies for glomerular disease?
Very good question. Two answers. Number one, there is a one size fits all theory behind it, because mechanical pressure propagates all glomerular diseases totally regardless of an underlying injury. That's why it helps for all. And this is part of the mechanisms we eventually revealed with our molecular studies. Number one. Number two, of course, we still need to be looking for specific interference and treatments of specific glomerular diseases. If you, for example, have a highly inflammatory glomerular disease or ANCA vasculitis, it doesn't have just to apply SGLT2 inhibitors. You just lower the mechanical pressure, but you need, of course, then to attack the underlying key injury mechanism. So it's the one thing, a RAS blockade and SGLT2 blockade is more a general supportive treatment for all, but of course, regarding in the underlying disease, we need to look for specific injury reasons and specific treatments in response to these injury mechanisms.
Okay, thank you. Dimitra Bacharati, excuse me, is asking the question: what is your opinion on the alternative view of albuminuria proposed by Russo et al. That states that albumin is normally filtered from the glomerulus in high levels, but it is the proximal tubule that reabsorbs it, so normally there's no detectable proteinuria. And she includes, as you explained for the slit diaphragm, we have the impression that laboratory investigations can have false positives and negatives, and thus can be interpreted in many ways. So,
yeah, exactly.
What's your opinion?
This is, yeah, wonderful question. Thank you so much. And I think the second part of the question answered already the first part. So basically, these observations are coming out of the early times of two-photon microscopy, which was in vivo live imaging. Two-photon in vivo live imaging was super exciting. And then albumin tracers have been used and was found, oh, wow, all these traces appear in the Bowman space very quickly. And based on that, there was an estimation of a very high albumin filtration coefficient. These studies could later on, with more advanced two-photon technologies, never be confirmed. And also when inhibiting endocytosis in the tubular machinery, it could never be confirmed to this extent. So, and number three, the at the time proclaimed massive transcellular albumin retrieval pathway could never be found. So I think after 15 years after this exciting hypothesis, and it was really good and challenging at the time. And don't get me wrong, based on the first in vivo results, it was the right conclusion to make. But I think 15 years later, with more advanced techniques, we know that it's incorrect, the hypothesis. There is no massive salvage pathway, while there is still a bit more leakiness of albumin all the time, which is then being reabsorbed. But this massive, basically a barrier which is non-restrictive to albumin, this is off the table.
Okay.
And this comes actually to a second question very nicely, how laboratory results, even being done in the best manner and analyzed and interpreted in the best possible manner at the time, can lead to wrong assumptions. That's why, you know, we need to continue working. And some of the things that I mentioned today might be seen in a different light 10 years ago. So I think this is why we need to keep going and look at things very carefully.
I have a question, a very short question, but I think you can talk like an hour or an hour and a half about this. From Abdelhamid Hamdi Ramazan, who asks, why can't podocytes regenerate?
Yeah, it's a good question. So philosophically talking, it's a known phenomenon in the human body and a known phenomenon of biology. The higher differentiated and elaborated structures are, the less capacity for regeneration they have, which is true for neurons and podocytes. And if you think how complex they are intertwined with the whole active cytoskeleton and their network, this is not a composition being made up for rapid regeneration. Now, there has been a debate, and a controversial debate, whether there is at least to some extent a minor regeneration. We have never been able to observe it, which doesn't rule out that under specific circumstances, there can be maybe a minimal, and we know that in the cleft between parietal cells and podocytes, particularly during development, there can still parietal cells moving in and form podocyte. So there might be a very minimum capacity, which doesn't play a role in adult humans. This would be my interpretation. And that's why glomerular diseases are so traumatic and the main reason for end-stage renal disease, why the tubular compartment can regenerate much better.
Okay, thank you very much. I'm looking at the question box again, but the question box is very small, so I have to. Let me see. There are a lot of people who are complimenting you on the fantastic webinar. There's another question which asks for what are actually the functions of the podocyte nucleus, or maybe you could take it a little bit broader and the podocyte cell body. As opposed to the food process.
Good and cell biological function. Of course, the nucleus, of course, as all nuclei, is there for the transcriptional process and securing the chromosomal message. So I think this is kind of almost generic. But why is the podocyte cell body so large? At least we can tell there's a lot of apical surface, which is important for the charge, which might also contribute eventually in part to filtration mechanisms. And it's important for generating all the nucleus, and the cytosol in the big body is responsible for generating all the cytoskeletal components that are needed in the food processes, as well as the diaphragm components that are then all being transported along these large processes. So basically the body needs to maintain a large network of primary and secondary food processes, and everything needs to be synthesized in the cytosol around the big cell body and the nucleus. This may be the best answer I can give right now. I guess there might be much more answers to this.
Yeah, there could be even more detailed answers about that, but,
Yeah.
but I think we should leave it at this. I'm looking if there are any other questions. If not, I would like to thank you again very much for the very great overview and enthusiastic presentation. I would like to thank everybody who attended today for their attendance, and I would like to ask your attention again for our next ERKNet ERA webinar, which will be on the 27th of June, as I think I'm correct. You might have a slide on that as well somewhere, but at least there will be a presentation by Paola Romagnani on the immune glomerulopathies, pathogenesis and treatment-oriented approach for clinical management. So I hope to see everybody again at the 27th of June. Wish you all a very nice afternoon, and thanks again, Tobias, for the nice presentation.
Thank you much, Joel. See you around. Bye-bye.
Thank you, everybody, also from ERKNet.